Good afternoon, everyone. I am Janaya Di Bornalo, and I'm going to discuss about the non-oxidative phase. The non-oxidative phase is also called the reversible second phase. During non-oxidative phase, pentose phosphate is converted to intermediates of glycolysis. The non-oxidative phase of the pentose phosphate pathway is broken down into four steps: the first process, second process, third process, and the fourth process. In the first process, we are going to take in the ribose 5 phosphate or the pentose sugar that is produced in the oxidative phase, which is then converted into ribulose 5 phosphate, which is an isomerase version of it using the phosphopentose isomerase. The ribulose 5 phosphate undergoes the second reaction with the use of the phosphopentose epimerase, and its product is the cellulose 5 phosphate. The only difference of the two molecules, the ribulose 5 phosphate and the cellulose 5 phosphate, is their stereochemistry. The stereochemistry of their third carbon is different. And why do we want to use the stereochemistry of the cellulose 5 phosphate to the second process? It's because the enzyme of the second process will only going to use the stereochemistry of the cellulose 5 phosphate. The cellulose 5 phosphate will be used in the second process and in the fourth process. In the second process, we will take in again the ribose 5 phosphate and the cellulose 5 phosphate. This process will use this transcatolase enzyme, which requires a cofactor called the thiamine pyrophosphate. What the transcatolase enzyme does is that it takes in two carbon groups from the cellulose 5 phosphate and then transfer it to the ribose 5 phosphate so that the ribose 5 phosphate will be extended into two carbon groups, and that is now what we call the pseudoheptulose 7 phosphate. After that, we will take two carbon groups again from the cellulose 5 phosphate, changing it to glyceraldehyde 3 phosphate. In the third process, we will use the products from the second process, which is the pseudoheptulose 7 phosphate and the glyceraldehyde 3 phosphate, as reactants in the third process. But in the third process, we are going to use a different enzyme, which is called a transaldolase. What the transaldolase does is that it takes three carbon sections from pseudoheptulose 7 phosphate, changing it to erythrose 4 phosphate. The three carbon sections that we get from the pseudoheptulose 7 phosphate is transferred in the glyceraldehyde 3 phosphate, which is now formed into fructose 6 phosphate. And this fructose 6 phosphate is one of the final products of the non-oxidative phase. In the fourth process, we will use the same enzyme in the second process, which is the transcutolase. We will use again the erythrose 4 phosphate, which is a product in the third phosphate, and again, what I've said earlier, we will use the cellulose 5 phosphate that comes from the first process. It is almost similar to the second process. We will take in two carbon groups from cellulose 5 phosphate to, to create fructose 6 phosphate, and then we were going to take two carbon groups again from cellulose 5 phosphate to form glyceraldehyde 3 phosphate. This is the net reaction of the process. So as we sum up all the four processes, this is what we actually get. 3-ribose 5-phosphate. The 2-ribose 5-phosphate comes from the first process and the other one comes from the second process. And what we actually get is 2-fructose 6-phosphate plus glyceroldehyde 3-phosphate. These two are the glycolytic intermediates that can be transformed into glucose 6-phosphate. And that glucose 6 phosphate can undergo the oxidative phase to form even more NADPH molecule. So when the cell actually means the NADPH much more than it actually needs the ribose 5 phosphate, we undergo the non-oxidative phase of the pentose phosphate pathway to help us generate even more of these NADPH molecules. On top of that, what the oxidative phase of the pentose phosphate pathway allows us to do is it allows us to actually ingest ribose sugars into our body, because via this particular pathway we can actually break down the ribose sugar into glycolytic instruments, and then those intermediates can be used via glycolysis to actually generate high energy ATP molecules. So we see that the link between glycolysis and the pentose phosphate pathway is the non-oxidative base of the pentose phosphate pathway. Significance of HMP shunt or the hexose monosphate pathway. HMP shunt is unique in generation. Because it generates important products like pentose and NADPH, which are actually needed for the biosynthetic reactions and various other functions. HMP shunt is also used in the synthesis of sphingosine and cerebrosides. Sphingosine is actually essential for cell growth, survival, migration, immune cell trafficking, angiogenesis, inflammation, and cancer. The next one is the importance of pentoses. Pentoses are used for nucleic acids, ribose for RNA, and deoxyribose for DNA. It is also used in the coenzyme synthesis. Coenzyme molecules are actually mostly derived from vitamins. They are also commonly made from nucleotides such as adenosine triphosphate and coenzyme A. Pentoses is also used in the synthesis of certain vitamins like B2 and B12. Importance of NADPH. NADPH is required for the biosynthesis of fatty acids and steroids. They also used in the synthesis of certain amino acids involving glutamate dehydrogenase, detoxification of drugs and foreign compound, and it requires NADPH. NADPH is also essential to maintain the integrity of RBC membrane and to keep iron ferrous state. Glucose 6-phosphate dehydrogenase deficiency or G6PD. G6PD deficiency is an inherited sex-linked trait. As it is linked to the X chromosome, most people who suffer from it are male. Sufferers cannot make the enzyme glucose 6-phosphate dehydrogenase. This will mean the circulation of sugar in their body is different. Deficiency is seen in all cells of the body but more severe in RBC. G6PD deficiency offers natural protection against malaria as the pathogenic parasites require reduced glutathione for their growth. Individuals with G6PD deficiency are at risk of hemolytic anemia. Hemolytic anemia is a disorder in which red blood cells are destroyed faster than they can be made. Symptoms of G6PDH deficiency includes black urine. And the second one is jaundice. It is a condition in which the skin and the white of the eyes turns into yellow. Third one is reduced glutathione.